So good evening, everyone, and welcome to the Seymour Marine Discovery Center's virtual Phytoplankton lecture, Small Things Considered, How Phytoplankton Make Life Possible. My name is Jen, and I will be the host for today's program. I would like to take a moment to share the UCSC land acknowledgement. The lands on which we gather is the unceded territory of the Awaswas-speaking Yupi tribe. The Amamutsan tribal band, comprised of the descendants of indigenous people taken to Mission Santa Cruz and San Juan Bautista during Spanish colonization of the Central Coast, is today working hard to restore traditional stewardship practices on these lands and heal from historical trauma. Tonight, our speakers will explore the histories, the presence, and the future of phytoplankton. These fascinating organisms cause beautiful blue luminescent oceans or deadly red tides. We'll learn about the current state of phytoplankton in Monterey Bay and around the globe. And we'll also understand how climate change is affecting these microorganisms that are critical to all life on Earth. We have three wonderful speakers this evening that will be sharing their expertise with us. Steve Mandel, Dr. Rafe Kudela, and Allison Cusick is all here this evening to share their research with us. I'll be introducing each of them more in depth as the night goes on before they formally start their presentations. And we have a few housekeeping items right up top, just so to make sure that everyone has the best viewing experience possible. So we do have closed captioning enabled on tonight's webinar. So if you would like to have a live transcript during tonight's presentation, make sure to turn on that feature down using the button either at the top or the bottom of your screen to enable closed captioning. We are recording tonight's lecture, and tonight's lecture will be posted onto the Seymour Marine Discovery Center's YouTube page within the next few days. So you can always access this presentation at a later date. We'll also be hosting a moderated Q&A after tonight's speakers present their findings. So please make sure to add any questions you may have to the Q&A feature, and we'll be able to answer as many as possible in the time allowed at the end of the presentations. And lastly, tonight's lecture is free with a suggested donation or membership to the Seymour Marine Discovery Center. So I'm really happy that you are all joining us this evening. And if you are in the position, I ask that you please consider either donating to the Seymour Center or joining us as a member so we can continue these very exciting education programs. I am going to be dropping these links in the chat. So if you're interested in learning more, you are welcome to head to the Seymour Center's website to learn how to get involved with us. So without further ado, I am really excited to welcome our first speaker this evening, Steve Mandel. Steve Mandel is a longtime participant in scientific research, science education, and wildlife conservation. His efforts have covered a wide range of scientific activity, including co-authorship of academic papers in the Astrophysics Journal and Sky and Telescope magazine, and founding of the Lions of Gur Foundation, dedicated to the conservation of Asiatic lions in India. An award-winning wildlife photographer, Steve has work juried for public display at the Smithsonian Natural History Museum and appearing in publications such as Smithsonian Books and the New York Times. He is the designer of an underwater 3D virtual reality camera system used by Nat Geo and by his Oceans 360 program to bring compelling virtual reality video to students and the public. As executive director of the nonprofit Oceans Micro, he provides marine microbiology programs to the public and to high school and middle school students. Steve is also a member of the UC Santa Cruz Division of Physical and Biological Sciences Dean's Advisory Council. Steve, I would love to welcome you to the virtual platform and have you take it away.
Well, thank you so much, Jen, and welcome everybody. And I got hooked on phytoplankton, and in particular, one type of phytoplankton, diatoms, after I volunteered for Allison's Fjord Phyto program in Antarctica, and I collected phytoplankton for later DNA analysis in her lab. And she's going to tell you more about that a little bit later this evening. And as I started to learn about phytoplankton, I really was just amazed at how interesting they are, how beautiful they are, and how vital they are to the survival of life on Earth. I really had no idea. Now, I'm a photographer at heart, so I started photographing diatoms. And the photos that I'm going to show you tonight, including the one behind me here, I took using various photo microscopy methods, including a complicated polarized lighting technique called differential interference contrast. And that's as technical as my language is going to get tonight. And my goal really is to produce photos that generate emotions and generate discussions about science and the effects of climate change, and ultimately the conservation of these very, very critical microorganisms. So tonight, we're going to talk mostly about two significant types of phytoplankton, dinoflagellates and diatoms. And in my talk, I'm going to mainly discuss diatoms, which have a huge impact on the planet. And Rafe Cadella is going to be talking more about dinoflagellates a little bit later. But let's first talk about what is phytoplankton anyway. Well, they are single-celled creatures, and the name comes from the Greek phyto, meaning plant, and plankton, which is wanderer or drifter, because they drift where the currents take them. But they're not really plants, and they have some characteristics of animals. So they have their own classification. They're called protists. And plankton are found in oceans, in freshwater, and in other wet environments like the cloud forest in Ecuador, where this diatom called Ulnaria came from. And on this diatom, you can see, because when I got this sample from some biologist friends, it arrived and it was still alive. And you can see the chlorophyll within the sample there. Those are the green clumps. Now, about 99% of diatoms and about 50% of the dinoflagellates use photosynthesis to generate energy. Now, before we jump deep dive into diatoms, let's take a quick glance at dinoflagellates. Now, the walls of dinoflagellates, and you'll see that in a second, a little video I'm going to show, are mostly made of cellulose. So we don't have extensive fossil records of them because the cellulose breaks down. And dinoflagellate in Greek means whirling whip. So most of them have these two flagella, and you can see it in the video coming out of the end, moving it around. But they're microscopic. So we're not talking about being able to swim against currents or tides. They can move around in their own little microscopic world. And we think they use these to maneuver around to get nutrients if they need it, or we're not 100% sure why. Maybe it's a defensive mechanism. And I bet some of you are already aware of dinoflagellates if you live near the ocean, or you've been to an ocean and you have seen either red tides, or as scientists call them now, harmful algal blooms, or HABs. And you're going to hear more about those later. And because they cause those terrible red tides and the problems with that. And they're also responsible for a lot of the beautiful blue luminescence that we see sometimes along the coast here and other places around the globe. So tonight, like I said, I'm going to focus on diatoms in my portion of the talk, because they make up a significant portion of the biomass of the entire planet. And they comprise 50%, 50, of the biomass in the oceans. So diatoms, and here's some photos I've taken of different kinds of diatoms. They have a top and a bottom, or as they're called by biologists, frustrules, and it's two interlocking halves, kind of like a box in its lid. And this next part is just crazy to me. Diatoms are made of silicon dioxide, which is glass, and they have evolved over 100 million years, these really remarkable designs that make them incredibly strong. And because of this ruggedness of design, we have intact diatoms that are many, many millions of years old. And I'm going to show you some of those this evening. Now, diatoms first appeared somewhere between 150 to 200 million years ago. And so they've been around quite a long time. And there are a lot of species of them. The estimates are that there's between 20,000 up to 200,000, some people say even more, species of diatoms. Minimum of 20,000, And, I think with mammals, there's like 6,400 species. I know of dinoflagellates, there's about 2,500 species. So this is a big, widespread, diverse creature. It really is. So now, when the organic material within the diatom dies, the weight, even though they're very tiny, microscopic, of the frustule, the glass house they're in, it causes them to settle on the ocean floor. And this has been going on for 150 million years. So in some places, the ocean floor is a half-mile thick with diatom frustules. And these deposits on land, which was formerly a seafloor, when it's there, we call it diatomaceous earth. And you have undoubtedly encountered this in a number of different products in different ways. Now, remember I said that diatoms, they use photosynthesis for energy. So they intake CO2 from the atmosphere, and they're absorbing a lot of CO2, which is great in this time when we have too much of it, like 30% of it out of the atmosphere. And they use the carbon for energy. They break apart the molecule and they release oxygen. So by eating all of that CO2, you might say they are leading the fight on climate change. But we're still falling behind, even though the diatoms are doing their best. So yay, go diatoms, keep it up. Now, the other thing they're doing, remember, they're releasing the oxygen. So they're releasing 50 to 70% of the oxygen that is produced on the planet every year. Now, much of that oxygen stays in the ocean, but a portion of it is released into the atmosphere. So the next breath, I almost wanted to sing Every Breath You Take, but I'm not going to. Your next breath will have oxygen that came from a diatom in it. It's part of what we breathe. It's part of how we live. Not only all of that, but diatoms really are at the base of the food web. They are the platform for life, and all creatures in the ocean and ultimately on land depend on them for their very lives. And not only all of that, and they're so important, but I think they are the most beautiful microorganism on Earth. So there's a lot to like about diatoms. So let's look at some diatoms from 30 million years ago. Now, these diatoms were collected in the mid-1800s in a place called Oamaru, which is on the South Island of New Zealand. And it's a very famous location for fossil diatoms. Now, in the UK in the 1800s, the Victorian era, science was becoming very popular. It was the age of Darwin. I think Origin of the Species was published in 1859, so right in the middle of that century. And it was the age of incredible drawings that came from a German scientist, Ernst Haeckel, drawings of diatoms and other microscopic creatures. He almost didn't become a scientist to become a landscape painter, because he was a great artist, but he painted, fortunately, these wonderful creatures. And like in this slide, what you see here is arranged diatoms, because what happened is that as science got popular, and another thing was going on, the production of microscopes increased, got more efficient, and the price came down, and people started buying microscopes. That created a big market for prepared slides. So people could bring slides home, put them in the microscope, and see all kinds of things. And one of the things they were interested in were diatoms and some of the other single-celled organisms. They made many of these, and they made them extremely well. So they have survived 150 years. And I bought a number of them, including this one, on eBay. And slide makers would hire artists to arrange the diatoms like they did with this one. And it was done with cat whiskers. Don't tell me how they got the cat whiskers. Cat whiskers and stretched glass rods. They heat the rods and stretch it out to microscopic points. And they had special microscopes where they could make these amazing arrangements. So many thousands of these slides have survived. And as a matter of fact, at the California Academy of Sciences in San Francisco, they have a huge collection of diatom slides, actually the third largest collection in the world. And I'm working with them now to photograph 100 to 200 of their slides for a possible future exhibit or a book. Now, here is a single diatom from the Oamaru deposit, and it's called Kittitonia elaborata. And to me, this is one of the most fantastic diatoms. This diatom that you're seeing here is a 30 million year old fossil. It did not survive the rigors of evolution. And I photographed this from a slide from the Cal Academy. So thank you, Cal Academy, for letting me photograph this. And this diatom is distinguished by these two processes, as they're called, extending out from it. You can see that, very unusual. And it's thought that perhaps they formed colonies and they used these to hold on to one another, sort of like diatoms holding hands. I wish it was Valentine's Day because that would have been fun. But you get the point. They were able to link up and hold on. Now, the next slide I'm going to show you is another example from the same location, 30 million year old diatoms. But instead of a single diatom, this is called a strew, which is just a bunch of diatoms on the slide. And I applied that very complicated polarization technique to get the colors, but mostly the detail and some of the structure. And you can see it's so gorgeous. And you can see why I'm so smitten with the great beauty of these single-celled creatures. Well, we've been looking at things from 30 million years ago. Let's come forward in time to about 10,000 years ago and take a look at these. Now, this is a sample. These were freshwater diatoms that came from the Hudson River Basin. And about 10,000 years ago, glaciers were starting to recede during that ice age and cut out the basin, and things were flowing, and these diatoms were around then, and they were left in deposits there. And they were alive at the end of the last ice age. And I was reading about this, and I realized these may have been alive, or probably were alive, when mastodons were walking around upstate New York. There may still be a few there, I'm not sure. But I just find relating the geologic age of some of these fossil diatoms and see what's going on the Earth is really fun to do. Let's come forward again, and now to about 4,300 years ago, which was like 2300 BC. The Romans still hadn't gotten into Britain or Ireland, so it was relatively untouched. There were people there at that time. And this slide, I mean, look at this. This is amazing. This comes from a place in Northern Ireland called Tomb Bridge. And these are freshwater diatoms. And just look at the abundance and the beauty of this. And this was actually collected from the soil right around this area in Northern Ireland around 1855, mounted very beautifully on a slide. And here we are in 2022 looking at a slide made 150 years ago of diatoms that are 4,300 years old. So it's really a lot of fun and very interesting, and you can learn a lot as you get into this. Now, this slide, also out of the Victorian era, is really interesting and one of my prized possessions because this slide and these diatoms were gathered by a British ship, the Challenger, from the Challenger expedition in around 1874. And this was almost a 4-year scientific expedition sponsored by the British government. And it's thought of as actually, some people say it was the formal beginning of oceanographic research. So this ship was sent out just to do research around the oceans all over the globe. And this sample comes from the Antarctic seafloor. So they did go down to Antarctica in the Southern Ocean, and on the ship they had literally miles of cable that they could lower down, and they drop a bucket, a dragging bucket for the seafloor along the bottom. They pull it up. This was pulled up from 11,700 feet below sea level. So they dropped this bucket down more than 2 miles, pulled along the bottom, and pulled up these diatoms. And I've showed this to Allison, who's an Antarctic diatom expert, and she recognizes, I think, most, if not all, of the diatoms in there. So let's jump one more time here. Speaking of Antarctica, this is not a Victorian slide because this is a modern slide from the waters of Antarctica. And this was gathered by Allison's lab. And you know, when I saw this, I thought, oh my gosh, this looks like the Death Star in outer space with snowflakes. So just kind of an amazing looking slide. You know, I said the last one was taken from 11,700 feet down. This one was taken from probably 8 or 10 feet down. You just dip a plankton net in the water in Antarctica, and I've done this, It's really fun. You pull it up, take it back, and this is what was found. So just a comparison between one in the water and one from the bottom. Okay, take a look at this. First, when I first looked at it, I thought, well, this is like a tree, but it's actually a microscopic collection of diatoms living on a very tiny bit of plant debris. And this is the diatom Licmophora flatabella. So the genus is Licmophora, species flatabella. It's actually a colony of diatoms that cling to each other, and they secrete this kind of mucousy stuff to hang on to each other. And they do so in these fan shapes. And I had no idea something like this existed. And I just thought this was so stunningly beautiful. I mean, it's beautiful and it's helping us breathe. So I'm just in awe of these. All right. So here's some other diatoms that sometimes they will cling to things. So these diatoms are on a small piece of kelp. Now here it looks pretty big, but if I hold a slide up, a microscope slide, you wouldn't see anything. You've got to magnify this 200 or 400 times to be able to see some of these things. Most diatoms are floating around freely in the water, some attached to things. And there's some weird diatoms I don't have time to go into tonight, but there are some species of diatoms that only live on the backs of manatees in Florida. The genus is Terciacola. There's some that live on sea turtles. That's the only place they live. Those are very rare, but there's a wide variety with over 20,000 species. You can imagine the diversity of these creatures. So this is the last Victorian era slide I want to show you. And this one just kind of blew me away. So this is an arrangement of 4 arachnodiscus, arachnoid, like spiders, named after, the spikes going out here. And this was put together in mid-1800s. And when I put this slide in the microscope and I passed polarized light through it, the nature of the way the glass, remember these are glass, the way it refracted it, kind of like a prism, they all bend the light differently, and it absorbed certain wavelengths of light and emitted others, and it leads to this image, which as soon as I saw this, it made me think of the beautiful stained glass windows in Sainte-Chapelle in Paris. And if you've ever been in there, they're so detailed and beautiful when the sun hits them. It's amazing. And that's what these made me think of. So the Victorians did something quite nice with these slides. They really did. But what about now? So now I want to go local. We just were global. Let's go local. And the next 3 slides are slides that I made from diatoms that were captured here in Monterey Bay. And typically I would go out to the Santa Cruz Wharf and drop my plankton net and bring them up. And so here's the first one I want to show you. And this one is called Catoceros. And it's another diatom that forms colonies. And you can see the individual cells, the single cell creatures, and they band together. And there's over 400 species just of this one genus. And the thing that looks like legs or antenna that are coming out, they're called setae, and they're not legs, they're not antenna. We think they help with buoyancy and possibly defense with this species. Some species of Catoceros are able to actually push chlorophyll out, kind of extending, their solar panels, if you will, to be more efficient at photosynthesis. Now here's another one from Monterey Bay, and this is Eucampia. And this also exists in a colony, and this was live when I photographed it. So you can see all the chlorophyll in each of the individual Eucampia pieces. And I've seen these, and they form a circle, almost a perfect circle. And they're quite lovely. And I put my slide together, and I'm not sure what happened, but I was letting the slide dry or let it sit too long, but it formed bubbles. And I found this piece of Eucampia on top of a bubble in my slide, and it made for a very pretty photo here. And this is the last local one that I want to show you. And this really fascinates me because this came out of less than 1 drop of water from Monterey Bay. And there are at least 6 different types of diatoms in here. And as I started doing this and pulling these diatoms out, I realized that the ocean is this vast, rich soup teeming with life. And I always thought of the megafauna, the big fish, and the mammals and the whales and all that. But every drop of water in the ocean is teeming with life. So keep in mind always that life in the ocean is seen. We go out whale watching, it's fantastic. And if you take a drop of water, put it under the microscope, it's also fantastic. So this is my last slide, and I'm going back to a Victorian slide because it's so beautiful. And there's some sponge spicules in here, but mostly diatoms. And as you've seen tonight, these are amazingly beautiful, and more importantly, they're stunningly important to life on Earth. And of course, we have to take care of the oceans, not only for the whales and the otters that we love so much here in Santa Cruz and Monterey Bay, and the fish and us. But also, please don't forget for the unseen life, the microscopic organisms like the diatoms that make all of this possible. So that's my part of the presentation, and I'm going to turn this over back to Jen and to Rafe Kudela.
Great. Thank you so much, Steve. I don't know about you, but I think I could stare at those photos that you shared all day long. They are absolutely mesmerizing. So thank you so much for sharing a little bit of insight into this teeny, tiny, complex world. Okay, well, I'm really excited to introduce tonight's next speaker. So Rafe Kudela has conducted aquatic ecology research for more than 2 decades. His research focuses on the factors and processes linking phytoplankton productivity to events occurring up the food chain, such as harmful algal blooms that affect crab fisheries. His work spans marine systems, the land-sea interface, and freshwater systems, and encompasses mitigation and prediction of harmful algal bloom events, changes in global productivity and fisheries, and linkages to human use of aquatic systems. Kudela has served as chair of the Global Ecology and Oceanography of Harmful Algal Blooms, also known as HAB program, and vice chair of Global HAB. He is an ex officio member and former co-chair of the U.S. National HAB Committee, and he serves on the executive committee for the Central and Northern California Ocean Observing System, also known as CENQUS. So Rafe, I'm very excited that you're here this evening. I'll go ahead and turn it to you, and you have the full virtual platform. Uh-oh, Rafe, it looks like we got you on mute over here.
Sorry about that. Try again. And so I was just saying it's exciting to be here, and I appreciate all of you attending and opportunity to tell you about why you should care about the small little creatures that are in the water. And so Steve was just talking about Oamaru in New Zealand, and this is the top of that same island. This is the top of the South Island. And this is a pretty common color of water for New Zealand. And if you ask most people, what color is the ocean, they would tell you blue. But of course, if you live near the ocean, it has many, many other shades. And so in my little part of this talk, we're going to sort of zoom out from a satellite view, and then we're going to zoom back in and look at exactly what's in that drop of water. And I'm going to introduce some new technology that's really revolutionizing our ability to understand what these organisms are doing. And so just to start, if you think about the color of water, the color tells you a lot about what's going on in the ocean. And so on the top, where you see those kind of funky looking tiles, that's an exhibit that we helped develop at the Exploratorium in San Francisco. And it allows people that are visiting the Exploratorium to actually do what we do, to look through the individual holes and find the color that they think matches. And so color is pretty easy to measure. And so it's one of the longest records we have, along with clarity, of the changes in biology in the oceans. And so we can look at climate change, for example, by examining how the color has changed. Of course, these days, you can do everything on your cell phone. And so on the bottom left there, there's an app that will actually help you. You can take a picture of the color and it'll give you some information. And one of the things that we do is every week we go down to the Santa Cruz Wharf and we are looking at the phytoplankton and whether there's harmful algae there, but we also look at the color. And so you can get an idea of just some of the colors there. And this slide shows you a more representative. This is about a year's worth of colors from the Santa Cruz Wharf. And you can see that just this amazing palette. Sometimes it's a deep blue, sometimes it's a greenish, sometimes it's red or orange, sometimes it's brown, sometimes it's lighter, sometimes darker. And that's telling us something about what's going on in the ocean, because the phytoplankton are a big driver of that color. And so some of the things that we're really interested in, and kind of stepping back from looking at it under the microscope, and I can't compete with Steve and his beautiful photos, so I won't try, but instead I'll tell you, why we're actually interested in looking at phytoplankton and counting them. And some big reasons are, are the oceans changing? And if so, why? Well, I think we all probably know that the oceans are changing, but the why is a difficult thing to answer sometimes. Oftentimes we want to know, well, exactly what's in the water, and is it good or bad for you? Is it going to kill you, or is it healthy and supporting, fish and whales and all the other organisms that we love? And then a big question that we've been asking is, you can take these beautiful images, but if you want to take images of lots and lots of phytoplankton, you get tired of it pretty quickly. And so is there some way that we can take advantage of machine learning and automation to have something do that for us, so we can just get more and more data to answer these questions? And so I'll give you a couple of very short examples of that. And I want to start by thinking of phytoplankton as just another food. And so organisms are eating phytoplankton. It's the base of the food chain. And so even something like the blue whale ultimately relies on phytoplankton. We rely on phytoplankton. As Steve said, the oxygen you're breathing is coming from the phytoplankton. And there's even people that will happily sell you phytoplankton, such as this nice glass of probably spirulina. And it has lots of positive properties. And many people consume phytoplankton directly because it has those positive properties. And so if you could imagine that if we could zoom in on a phytoplankton and look what's inside it, we can develop this thing called a phytoplankton food quality index that tells us, well, how good is it? Is this, junk food, or is this a nutritious salad that you're eating? And so if you imagine zooming in on a dinoflagellate like we have there in the background, and there were a nutritional label on it, we could look at that and say, okay, the fat's not too bad. It's kind of high in carbs, but that's okay. And that's what we do when we go to the grocery store. Well, we can do something like that in the oceans as well by taking advantage of the fact that the phytoplankton evolved with different properties. And so to do that, what we can do is we can look at this compound called long chain essential fatty acids. These are compounds that are super good for you. They're very nutritious. And there's been a lot of work demonstrating that the more long chain essential fatty acids, or LCFA, if you want a shorter version, the more in your diet, the healthier you are. And so we see positive trends with things like shellfish and planktivorous fish like anchovies and sardines, that the more of that fatty acid they're getting, the faster they grow, the healthier they are. And so we can actually use that as an indicator of what's going on in the environment by looking at, is there a lot of LCFA, or is there not very much LCFA? The really nice thing about that is it's a function of evolution. And so the major groups of phytoplankton have different levels of LCFA, and it's less important what the environmental conditions are right now, and more important, which group of algae did it come from. And so I didn't bother to label all of them, but I'll just point out a few. The sort of bright blue that's close to zero at the bottom, those are cyanobacteria. And so the fact that it's so low, close to zero, it's basically like eating cardboard. And so really bad for an ecosystem. Dinoflagellates in orange and diatoms in that sort of purple color are really, really good. And so these are generally what we consider healthy phytoplankton. Because it's following these evolutionary trends, that evolution is also selected for different pigments, which gives us those different beautiful colors that we get with phytoplankton. And so we don't need to know every single species. And so Steve told you that, for example, within there's hundreds of, or even thousands of species within some of the genera. And it doesn't matter for this analysis. We just need to know what big group they're in. And so is it a diatom, or a dinoflagellate, or a chrysophyte, or a cyanobacteria? The really cool thing is we can actually get at that from the color. And so this is an example of San Francisco Bay, and it looks kind of crazy, like maybe you have a little too much wine or something, because we've taken a multispectral camera and we've looked for those individual pigments, and then I color coded it where you see lots of red, like in the salt ponds in the bottom of the image, that's dominated by dinoflagellates. Where you see a lot of blue, that's dominated by cyanobacteria. And in this case, where you see a lot of green, those are chlorophytes. And so we can actually take images of that from an airplane and convert that into the LCFA and tell you, well, is this good quality or is this bad quality for a particular time? And we did that for a fairly long period of time in San Francisco Bay. And here what you're looking at, the colorful bars are that plankton food quality index. And so the higher they are, the better the quality is. The black line is a drought index. And so you all know probably we're in a mega drought right now. And we go back to 2010, it was not drought conditions. And then the drought got worse and worse and worse. And as the drought got worse, the food quality dramatically decreased. And so by the time you get to 2016, basically there's not very much good to eat in San Francisco Bay in terms of the phytoplankton. It got a little bit better in 2017. And then unfortunately, because we're still in the drought, it's kind of dropped off again. And so we can actually start taking this information and tell you something about what's going on in the ocean or San Francisco Bay in terms of whether this is a healthy environment or a not so healthy environment. The problem is that we can get to things like diatoms and dinoflagellates and cyanobacteria, and that's super useful in some cases, but some of those can be dangerous. And so some examples here, Pseudonychia in the bottom left, that's a diatom, but it produces domoic acid. Alexandrium and Akashuo and Dinophysis and Goniolax, those are all dinoflagellates. They produce various toxic compounds. And then of course, cyanohabs, in addition to being poor food quality, they also produce some pretty nasty compounds that you don't want to be exposed to. And so to get to that level, we really do need to put it under the microscope, and we need to look at the individual cells and say, is this something that is bad for us, or is this something that's just a good food quality, a good organism that we're not too concerned about? And so the problem is that it's tedious. You know, you can put it under a microscope and you can look at the slide, and we do this. We do it once a week for the Santa Cruz Wharf, but it takes a lot of training to be able to identify all this phytoplankton, and it's not the most fun thing to do. You know, after 5 or 10 minutes, you're like, oh, those are really beautiful, but I'm tired of counting. And yet you have to keep going because we need the data. And so an obvious question is, well, given the rise of technology and instrumentation and things like machine learning, is there some way to automate this? And there is. There's a new instrument called an imaging flow Cytobot, which looks pretty boring when it's in the can. It looks like a big aluminum cylinder, but a pretty amazing device inside. And it's basically an underwater microscope, if you want to think of it that way. And so we've got 3 of these, the first 3 that were on the West Coast of the United States, actually. One we run in San Francisco Bay with the USGS cruises. The picture on the right is showing an instrument up at the Exploratorium. And then we have one at Santa Cruz Wharf that's operating pretty much all the time. And so just to give you a little idea of how this thing works, it's an amazing piece of machinery. And so it brings in a syringe of water. And so it pulls in a syringe of about 5 milliliters of water every 20 minutes or so. And then it forces it to go through in basically a single stream. And so the phytoplankton cells are going through one at a time. And as they go through, this instrument is going to interrogate them. And so what it's going to do is it's going to shoot lasers at them. And as those lasers go by, it's either going to scatter, which tells us something about the size, or it's going to cause fluorescence if it has pigments. And so we also get the fluorescence. And the detector can see that. And then the really cool thing about this, because this is basically the way they count your red blood cells if you go to the hospital, but what's different about this is it can also take pictures of every image. And so as things are going by, it grabs a photo. And so we have a digital record of everything that was in that 5 milliliter sample. And so this is what it looks like when we get those data back. And so you can see just the amazing diversity of images. We also have some heterotrophic organisms in there, some zooplankton, but it takes a picture of every single thing. And you might be wondering, well, why isn't in those beautiful colors like Steve uses? It's because for machine learning, grayscale is actually better. And so the amazing thing about this is once we have the pictures, we run it through a machine learning algorithm. It tells us the size, but it also identifies the organism. And so instead of me having to sit there with a microscope and say, well, that's something I'm worried about, that's something I'm not worried about, we can let the instrument do it for us. And it takes a sample every 20 to 30 minutes, basically as long as you're running it. And so you can imagine how much more data you get when you're using this method versus going down to the wharf, which is certainly pleasant, and grabbing a net tow, but you just don't get nearly as much information. And so to give you an idea of what we get from that, this is a mooring that Mbari put out that's actually wave powered. And so the power is being generated by the mooring to drive this instrument. And on the bottom, you can see that ribbon. These are some of the images that came off of it. And it's so much data that we can start sort of adding it all up and saying, well, what are the major groups of phytoplankton? And so all those names on the right there, those are things that we can identify automatically using machine learning. And so you can see, for example, that sort of deep red color, that was an increase in dinoflagellates in this particular case during the end of June of this last year. The other thing, though, we can do is we can identify those phytoplankton, and many of them are just good food quality. But the ones to the right, that's Alexandria, which causes paralytic shellfish poisoning. There's Dinophysis, causes diuretic shellfish poisoning, and there's Pseudonychia, which causes domoic acid poisoning. And so we can actually use this as an alert system where it can just basically send you a text message saying, "Look out! There's a lot of these potentially harmful algae showing up in the sample." And so the Ocean Protection Council, funded by the state of California, was interested enough in this that they're actually paying for about 12 of these instruments to be deployed along the coastline of California so that we can get this continuous data and both use it as a harmful algal bloom warning system, but we can also do things like look at the food quality, we can look at the biodiversity, we can look at how things change with an El Nino or a La Nina or a storm event. You know, the possibilities are endless in terms of what you can do with the data once you start collecting it. And so I'll just stop my little part of this talk here. And the next time you're down at the ocean, I really encourage you to look at it carefully and look at the color. And it's probably not blue. And the fact that it's not blue all the time tells us a lot about what's going on. And it's in large part because those little tiny organisms are in there and they're changing the color, they're changing the food quality, they're allowing the amazing life that we have here in the Monterey Bay to thrive. And of course, they're producing that oxygen that we breathe. And so if you're interested, that horrible link that has 128.114, that's the real-time data that's coming from the imaging flow Cytobot. And so you're always welcome to go there. And as soon as we get the data, it goes to the web and you can see what's going on in your backyard at the Santa Cruz Wharf. And so thanks very much. And I'll be happy to turn it over to Allison.
Awesome. Well, thank you so much, Rafe. I have so many questions lined up and ready to ask you, which I'm really excited to dive into. So that's a good reminder for all of us at home. If you do have any questions for any of our speakers this evening, please don't forget to add those to the Q&A for our Q&A portion at the end of the evening. Okay, so it looks like we are about to introduce our third speaker, who I am thrilled is joining us this evening. Allison Cusick is a polar phytoplankton ecologist. Her research in Maria Burnett's laboratory at Scripps looks at how melting glaciers might be influencing phytoplankton communities in coastal fjords along the Antarctic Peninsula. She aims to understand how phytoplankton responds to a changing world, looking through both an ecosystem-wide and a genetic lens. Allison obtains her samples and data through the NASA-funded citizen science program for Phyto, which she co-founded in 2016 with her advisor and polar guides to partner with tour ship operators in Antarctica. Allison received a dual master's degree in marine biology and marine biodiversity and conservation from Scripps Oceanography and earned her bachelor's of science in biology at the University of Washington. Through her Women Scientists blog and social media platforms, she inspires others to pursue careers in STEM fields that get them outdoors. So Allison, I'd love to turn it over to you, and I can't wait to hear all about your research.
Okay, thank you so much for that introduction, and hello everyone. Thank you for being here. I also have to say hello, Mom and Dad. Thank you for being here. Let me start sharing. Oh, and hello to all my friends and family. Okay, so I wanted to talk about, during my portion, some of the strategies that phytoplankton have developed to survive in this gigantic ocean. And I want to share an example, of course, of their mightiness that comes straight from one of my favorite places in the world to work, Antarctica. And I hope to convince you, if you haven't already been convinced yet, just how incredible phytoplankton are. And some of the information, of course, will build on what Steve and Rafi have already talked about. So when I think of the ocean and phytoplankton, honestly, my first thought before I got interested in this field was boring. But there's so much happening, and it impresses upon me how they've evolved strategies to survive in this watery environment that's incredibly spacious. So it was billions of years ago that a single original phytoplankton, the cyanobacteria, found a way to use sunlight to make energy and split water. And it just happens that oxygen's a waste product. They don't care about it. Thanks for us. And this is one of the strategies that allows them to evolve. You know, of course, you have to be able to eat in the ocean. You have to be able to reproduce in the ocean, and you have to be able to avoid death. So you've already been introduced to a lot of the concepts that go into these different strategies. But when you're eating light, a lot of people ask me, what do phytoplankton eat? One of them is light, and that is different photons in the visible light spectrum. And the red photons and the blue photons are incredibly tasty. And they've also evolved different pigments, like Rafi talked about, to harvest other photons when they need them, depending on light levels in the ocean. And you basically have to become an incredibly effective floating solar panel. You want to stay in the sunlit layers of the ocean. You don't want to go below where it's dark and you can't find this food. Also, you use carbon dioxide, so you can eat that from the water. And of course, you also need basic nutrients, just like you would put on your plants, fertilizers on plants in the garden. They need the same nitrates, phosphates, silica, and iron. That will come in important later in my talk. So if you can't float around to get enough light, then you might also develop swimming strategies, like Steve showed in that video of the dinoflagellate swimming around. And if you like sci-fi, I really think you're going to love phytoplankton even more, because if you are unable to make your own food, then some of these phytoplankton have developed strategies to eat each other. So you can, some of these dinoflagellates will cast a mucus net around a group of diatoms and dissolve them and slurp them up. Others will basically do something where they capture the photosynthetic solar panel superpowers from other phytoplankton when they eat them and then use them for themselves. That's called kleptoplasty. And so there's some weird strategies to get food in the ocean when you're a single-celled organism. So we have another strategy we have to figure out as a phytoplankton. We need to reproduce, of course. So you can do this asexually. So that just means that these phytoplankton can split themselves in two, and they can divide over and over and over again exponentially until they eventually run out of something that they need to survive in the ocean. And you might think that's a really excellent growth strategy, but now you've just cloned yourself. And so you don't really necessarily have good genetic diversity in your population. It's kind of like monocrops in agriculture. If you have no genetic diversity, you might be less resilient to any changes that come along in your environment. So these crazy phytoplankton also go through sexual reproduction. So this is just showing that diatoms and dinoflagellates have a life history where they can mix up their genetic pool by releasing gametes. And this allows them to not only return back to the normal vegetative size if they've shrunken down too small after dividing, but also to help the population become diverse. And also you've seen that satellites are monitoring the health of Earth and ocean color from space. And these blooms are so incredible that there's an image from NASA's Ocean Color that on the slide here that shows portion of Antarctica where this green color and blue color is being picked up as a peak through the clouds. So when they grow in such large numbers, that chlorophyll is what we're seeing. And this is not only on land, which is the green that you're seeing here, but also from, of course, these phytoplankton in the ocean. And you can see as the seasons change here between the northern hemisphere where it's summer and the southern hemisphere now is summer. And then winter comes, all that vegetation dies off. It's like the lungs of Earth breathing between terrestrial land plants and ocean microplants. And I just think this is mesmerizing. Seasonal change. So we have eating strategies, reproducing strategies, and now we have to avoid death. So how do we do that? You could imagine with all your siblings growing and you yourself eating all the nutrients around you, you're going to have nutrient loss. This can be a problem, so you could move around to different areas. But also the ocean has a lot of viruses floating around, and viruses are looking to attack phytoplankton. So through viral lysis, it is dangerous out there. So some phytoplankton populations kind of trigger like a cell death response, programmed cell death to kill themselves off. And researchers are studying this process to understand why it might be happening and what it does for the population. And also, this is, I think, incredibly crazy. If your environmental conditions aren't favorable for you, then you can encapsulate yourself as a spore and become a cyst that then sinks to the bottom of the ocean or to the sediments below and wait until conditions are more favorable for you to grow. And there was a group of researchers that tried to revive some cysts from old sediments that were 7,200 years old, and they were viable and germinated. So that, I think, time span wise, if you could basically kind of hibernate to protect yourself for that long, that's incredible. And then, of course, you have other organisms that are trying to eat and graze you from the population. So that's where you can develop spines and you can produce all these different toxins to kind of keep yourself from being eaten. And then, of course, finally, you are going to want to avoid sinking, and you can do that through increasing your surface area. You also don't want to have advection being brought out of that surface sunlight area, so you can flatten your body, you can grow appendages, and this allows you to increase your surface area without adding density, which would then make you sink if you were too heavy. And the last thing I want to share with you about their superpowers and their ways of evolving is that you have to watch out because they'll start hypnotizing people. And now I want to share how the more you get to know them, you'll be amazed and fall in love. And I want to share with you how I fell in love with these phytoplankton in one of the most extreme regions of the Earth, which is where I'm working on my PhD work and hopefully my career as a polar scientist. So Antarctica, something magical happens. Antarctica is, the icy continent. And in the winter, when there's no sunlight, the surface of the ocean begins to freeze. And that actually doubles the size of the continent when you're looking at an image like this. And that process is incredibly important because as soon as the sunlight returns after the winter season, 5-6 months long, you get this incredible sea ice diatom bloom. And I like to call this Antarctica's invisible forest because there's no trees on land. It's all in the water at a microscopic level. And these phytoplankton bloom under the ice and they connect that non-living world to the first link to the living world, because what comes next down in Antarctica are something called Antarctic krill. And krill are a type of zooplankton or animal plankton that are key species down in this ecosystem. And krill are then feasted upon by all of these big animals that come down to Antarctica or live down there, like the penguins and the seabirds, the seals, and the big whales, the baleen whales, blue whales, sei whales, and humpback whales, minke whales. And this is a classic example of polar food web. So it's usually a really short transfer of energy from a single phytoplankton to a krill to a giant animal. And this connection is also important because these giant animals poop, and that poop is full of iron. And so whale poop and seal poop and penguin poop is incredible for bringing iron back into the ocean that is then eaten by the phytoplankton. So that keeps that whole cycle going. And a lot of this is directly linked to the production of sea ice. Antarctica needs to freeze every winter. It needs to stay cold. And these krill, when they're young, feast on the sea ice diatoms. And then when they become adults, they move out into the open ocean and they can feed on other types of phytoplankton that live in the open ocean. And believe it or not, krill actually have a preference for the types of phytoplankton they're eating. You've seen already that there's a different nutrition content, so to speak. So krill, this is an awesome comic from the Scholarly Krill. And they say, oh, diatoms, they're my favorite. And this other krill is handing a bouquet of diatoms. I know they're in bloom. So the krill have been shown through researchers dissecting stomachs that they prefer these large, round, centric or chain-forming diatoms that are full of that lipid and fat and nutrition. And different environmental conditions, sometimes we see a shift to smaller phytoplankton like you see pictured here, that size difference between a large round diatom and a tiny small flagellate. And that could be due to freshwater inputs from melting ice. It could be just that it's later in the season and the diatom bloom is gone. That's one area of research that I'm particularly interested in looking at. And this doesn't just matter for the krill, it also matters for all of those marine animals that go through a planktonic stage during their life development. So it's also for the fish, it's for swarms of squid, it's for anything living in the ocean, crustaceans that have this larval stage and need to feed on phytoplankton. And then of course, you know, maybe you've heard of omega-3 from fish oil or krill pills. Just remember that the omega-3 is actually coming from the phytoplankton. And of course, I want to impress upon you too that there's seasonality to this. So it's not just in Antarctica, winter to summer, but it's also elsewhere in the world. So a lot of the spring time is when phytoplankton really get their start to the season. And then there's different trends that happen over the season. And so how would you monitor any of this? One way we're doing that in Antarctica, which the future relies on collaborations. The more collaborations, the more we can understand these giant regions of the world, but also remote regions of the world. And so in this sense, there's an active tourism community of ships that go down and visit every year. And so with the help of guides, polar guides and the Polar Collective, we train guides on board these tour ships to collect phytoplankton. So then when travelers come for their trip, they can participate. And this is exactly how Steve and I got to know each other. So one of the science boats from Fjord Phyto, phytoplankton of the fjords, which are common in areas that used to be covered in glaciers in the last maximum glacial ice age. Fjord Phyto take a phytoplankton net in one of the little boats and just collect that water, and then we can strain it off. We can preserve this sample to do DNA sequencing, so I can see maybe which species are there, but also genetically, how are they responding to the environment that they were taken out from. And then of course, we can look under the microscope with my colleague, Martina Massioni in Argentina, who can identify all of the individuals, calculate carbon biomass, how much food are they contributing to the ecosystem, and then I'll also get those counts that you've already heard can be quite tedious to do. So Antarctica is amazing, but it also has now one of the hottest temperatures ever recorded. This was in 2020. It was definitely, I was down there at this time, it was definitely t-shirt weather, and it reached 65 degrees. And scientists have known that Antarctica is one of the fastest warming regions in the world since the 50s, since there's been good data collected from that region. And of course, heat is not good for cold regions. So 97% of the continent's covered in ice. This ice is 90% of all the world's ice and 70% of all the world's freshwater locked up in snow and glaciers. And on the peninsula, which is the portion of Antarctica that I focus on studying, the glaciers on land, 87% of them have been retreating. So they've been losing mass. And there's that freshwater input that's coming into the marine coastal saltwater environment. So that is changing this environment where these phytoplankton live. And that can also, as I said earlier, change the different types that we see, change the sizes that we see. So how might that be affecting the Antarctic ecosystem? And when we think now, what about the rest of the ocean? How is climate change influencing oceans around the world? There's a phrase that I've heard that I really like, where we say the oceans are becoming hot, sour, and breathless. So that just means temperatures are rising, sea surface temperatures, also sour. So there's carbon dioxide in the atmosphere when it gets into the ocean that can change the chemistry, reduce the pH of the ocean. And that's not necessarily good if you're an organism that needs calcium, for instance, or if there's a phytoplankton that uses iron. Maybe some of the proteins that they use to bind iron to photosynthesize might not be working. This is research that scientists have been working on understanding in the lab and in the environment. And then breathless, of course, warmer. We all know a can of soda that's warm doesn't have as much carbonation in it. The similar effect is happening to the ocean. It's less able to hold the gases, oxygen, and also carbon dioxide. If the ocean's taking up carbon dioxide that would be in the atmosphere, that could be an issue. And then what does this mean for the phytoplankton? It's a hard thing to really tease apart, because of course you can do highly controlled lab experiments, but when you go into the actual environment, things could have a different answer. So we say there's winners and there's losers, and there's a lot of effort to be done understanding the diversity, the changes in the diversity of phytoplankton. But they're not just small scale organisms. They have big scale impacts, and it's complicated. And so the role in the ecosystem is so interconnected with all these feedback systems. And we can try to develop equipment, like you saw earlier, to understand this, and even in the water and at the level of satellites in space, which I think is incredible. And so how can we use phytoplankton superpowers to our advantage? I don't know if any of you have already heard of this idea, because it was attempted pretty realistically in the 80s, is to do iron fertilization of the ocean. So if you just took a ship and dumped a bunch of iron in the ocean, perfect. That would just let the phytoplankton grow, and then the phytoplankton would use all that carbon dioxide, which would bring it out of the atmosphere. The fish would eat it because there'd be enough food. It sounds like an awesome idea. When they die, they bring all that carbon down to the seafloor, and they will lay there for hundreds or more years. It's basically like an attempt to speed up the creation of fossil fuels, which we've innovatively learned how to extract for our societal benefit, well, to a point. And but we also have to remember that that is like an idealistic way of how things would work, and the reality could be very different. So you know, what people have learned from the coast after big harmful algal blooms come through is that it doesn't necessarily create this paradise environment. That the aftermath is that it's depleted nutrients, depleted oxygen, not a good outcome. And remember those cysts I talked about, all those cysts that might sink to the bottom when conditions are more favorable for a long time later? There's a lot to consider when we're thinking of these large scale fixes. And then the last thing I wanted to also talk about how we could use their superpowers is taking advantage of algae or phytoplankton's ability to make the fats, to store energy and harvest enough as a new green source of fossil fuel. And the need for energy is obviously increasing. And if algae could help meet that demand, it could be converted to a different type of fuel source that we could use. And there's a lot of biofuel research being done. I used to work with a team here in San Diego, funded by an oil company, to figure out which algae grow the best and which algal genes are involved in making them the fattest, and then genetically engineer those genes into the really hardy growing algae. To be a possible source that you could harvest through summer and winter, to create demand, or I mean to supply demand to the world. But again, there's challenges there. It's enough space to grow them, enough salt, enough nutrients. What if they get out? What if there's invasive airborne things coming into the ponds? But there's a high potential for literally green energy. So tonight we've shown you how inspiringly beautiful the invisible forest is, capturing imaginations for centuries, how integral they are to the food web, feeding different stages of life of marine animals, and how large numbers can become harmful, how they could be used for good, and how researchers are trying to understand everything about phytoplankton. So though they're tiny, they're mighty, regulating global oxygen and carbon cycles. And with that, I want to say, I hope the next time you hear about phytoplankton, think large scale. Thank you.
Absolutely wonderful, Allison. Thank you so much for sharing. I absolutely love the phrase tiny but mighty. I think that may become a new catchphrase down the line. So thank you so much. So we have a few minutes actually for questions. So I have a few questions lined up here for our speakers from this evening. So I invite all of our speakers to go ahead and turn on their video camera and unmute themselves, because I know a lot of questions came through the Q&A. Our speakers have been doing a really great job of answering those Q&A questions in real time. So definitely check that out as well. But there were some really interesting questions that I want to make sure our whole entire audience had the opportunity to hear. And so one of those really interesting questions was about ocean acidification. And so I know, Rafe, you went ahead and answered this in the Q&A, so I'm hoping that you can answer it live as well. But Maria asked a great question: if all phytoplankton are affected by ocean acidification, or just the ones that maintain their buoyancy with calcium carbonate shells. So if you could just answer that for our audience, and then also define what ocean acidification is too.
Sure, absolutely. So ocean acidification is a decrease in pH. And so it's that the ocean is literally becoming more acidic. And so normally under sort of before the Industrial Revolution, the ocean is slightly basic. And when we talk about acidification, we're not talking about it turning into lemon juice. It's relatively small changes in pH, but these organisms have evolved over millions of years with a pretty constant pH. And so it has a big impact. What it does to the phytoplankton is, yes, absolutely. If they're using calcium carbonate, that's going to dissolve faster. And so they have to spend more energy to keep that calcium carbonate in their shells. And so those organisms are very impacted. But all phytoplankton are impacted by acidification because it changes the availability of carbon dioxide in the water. And so for some of them, it's easier to get the carbon dioxide, and you can get blooms that you may not want because you've made it just easier for them to grow. For others, they've evolved not to use carbon dioxide directly. They're using things like bicarbonate, which is another form of carbon. And so those ones actually do worse. And then something that we're discovering relatively recently is if you start to lower the pH, we can have unintended consequences. For example, there's more domoic acid produced. And so domoic acid is what shut down the Dungeness crab fishery a few years ago. And so, you know, there's a lot of concern about acidification beyond the direct impacts, but there's all these potentially indirect impacts that we just don't understand very well.
Yeah, interesting. Wow. And so this next question is for Allison. And Allison, we were hoping that you could elaborate a little bit more of what you've been seeing in Antarctica with these warming temperatures. You touched on it a little bit in your presentation, but I'm hoping you can talk about what changes you are possibly seeing in the phytoplankton, whether it's the species makeup or sizes or just less of them. Just elaborate a little bit more about what some of the changes you might be seeing.
Yeah, excellent question. So I have personally only been looking at the Antarctic Peninsula for the last 5 years, but one of the major hypotheses or questions that exists that researchers down there are interested in is they've been seeing this shift. So when there's more fresher water or water coming from melting glaciers, they tend to see, researchers tend to see the smaller phytoplankton or flagellate plankton. So those cryptophytes are what they're called. And the issue with Antarctica is that it's so remote and difficult to actually monitor and get good seasonal data on. So a lot of the efforts are kind of like snapshots of the season of what's happening. And so one of the things that the program we're doing with the tour industry, Fjord Phyto, is that we can kind of now capture that November, the season starts in November and goes through March. It's the Southern Hemisphere summer. We kind of can get a little bit more of a a time series, so to speak, of what's happening as it's changing and at different locations all over the peninsula. And so one of the things we published in Polar Biology, the first results of the first couple of years was a couple of these blooms that were so large that hadn't been detected before. There's an unarmored dinoflagellate that hadn't been observed down there. So it's kind of like filling in the gap of what might be missing in this seasonal change. And then the other samples that we have that I'm looking at with the genetics, I'm currently in the process of analyzing. So that will definitely be out hot off the press when I finish my PhD. So stay tuned.
Yeah, I love it. Great. Well, now we can just keep tabs on your research. That's super exciting. Great. So just a few more minutes here, but Steve, we got a really interesting question asking from, I believe, Brandy, who was asking about if you have any tips of how you prepare your slides, because they're absolutely breathtaking and striking, and maybe belong in a museum, who knows. But if you can quickly share any tips that you may have for preparing these beautiful, beautiful slides.
It's not so much in the slide preparation, because I'm just taking a drop of water. I let the phytoplankton settle in a small amount, and I put in a pipette and put a little drop on a slide and drop on a cover slip. The secret is really with the light that you pass through it. So in a light microscope, there's different filters, polarization techniques. Like the slide behind me, I route the light around the side. It's called a dark field, so the back is light. But because these little gems are made of glass and they're like prisms, they all react quite differently to different lighting. And so I look for the best way to illuminate it, but it has to do not so much with making the slide, but how you light it up.
Mm-hmm. Gotcha. It's all about the light. All right. So before I get to my very last question this evening, Rafe, I have to ask you, what is that cost of the IFCB? Because you said there was only 3 on the West Coast. Like, how much was it, and how much is it to maintain?
So there's more now. There's about 10 on the West Coast now, but they're not cheap. And so if you're thinking about buying a new car, you could buy one of these instead. And so it's $135,000. However, they last at least 10 years. And so if you think of how much you're saving by not paying somebody to do the microscopy and just how much incredible data you get, it's actually really inexpensive. And for science equipment, the smaller the market, the more things cost. And so it's a pretty reasonable cost for things that we do in oceanography.
Yeah. Oh, interesting. Okay, so my last question I have for all of our speakers this evening is always one that I have on my mind, and I think a lot of our at-home viewers do too, is that how can we help? Like, how can we get involved, right? We just heard about all of this incredible research and work that the 3 of you are engaged in, but I'm curious, like, how can, like, me as a non-scientist, can help be part of a solution? So maybe, Steve, you could kick us off with your suggestions, and then we could go to Rafe and then end with Allison.
Well, I'll just tell you quickly about my experience, because the way that I deal with this, it's global warming, of course, is an overwhelming problem, but I decided to learn as much as I could and then educate others. So I have a nonprofit program, Oceans Micro. We're going to be doing some live programs starting in March at the Seymour Center. We do programs online for high schools here and middle schools here in the US. And we just started doing that online in Australia also. The timing works out. So for myself, I decided I would educate other people about this. And so that's my approach.
Yeah, definitely. Education is key. Rafe, what would you like to add to that?
Yeah, I would say definitely, support the organizations you love. And so Oceans Micro, Seymour Center, those are the groups that are getting the information out there, making it accessible to you. And I always tell people, if you're concerned about the environment and climate change and what's going on in your backyard, vote. And, whatever you vote for, as long as you're passionate about it. But if you don't vote, you're not making a difference.
Yeah, definitely. That's great advice. And Allison, would you like to bring us home?
Yeah, so I like to say to people, be like a phytoplankton. Any one phytoplankton might not seem amazing, but when they do something, anything in large numbers, that's when you have the most impact. So just to, second, since my program is citizen science in interacting with non-scientists and scientists, I would suggest look for a local project for monitoring algae around you. If there's volunteer efforts near where you live, if you do live near the coast, and if you don't, there's puddles and plants. Wherever there's water, there might be a plankton or algae for you to take a picture of. There's a lot of citizen science projects on scystarter.org is like an encyclopedia where you can type in phytoplankton and look. So if you had access to a microscope, there's some cheap, affordable options online now, or Foldscope is a paper origami inspired $1 microscope. Just take a photograph of a phytoplankton and upload it to iNaturalist, which it's a simple observation, but that's always the power of the crowd, or the power of many, is that the more people just looking around and caring about phytoplankton, the more ideas you'll have of how you can get involved. So
yeah. Well, definitely. Well, thank you so much to the three of you for sharing your time, expertise, and energy with us this evening. I can't thank you enough. And I know our virtual audience at home is also thanking you as well. So that is, I know we're over time. So thank you all for sticking with us on this Thursday evening. It's been an absolute pleasure. So again, this lecture was recorded and it will be posted to the Seymour Marine Discovery Center's YouTube page within the next few days. And please, please, please take what Steve, Rafe, and Allison said about how you can get involved and how you can help, because I keep using the phrase that Allison said, be like a phytoplankton, where we can do more good together, instead of just one individually. So thank you so much, everyone, for joining us this evening. Have a great rest of your day, and we hope to see you at the next virtual or in person, or maybe even a hybrid Seymour Center event. Take care, everyone, and have a good night.